Welcome to our deep dive into two fundamental study designs in clinical research: cohort studies and case control studies. These methodologies are essential tools for understanding the relationships between exposures and outcomes in medicine. By the end of this video, you'll have a clear grasp of how each design works, their strengths and weaknesses, and when to use them. Let's begin by setting the stage with why study design matters in clinical research, ensuring that the results we obtain are both reliable and meaningful for patient care. Choosing the right study design is crucial in clinical research. It determines how data is collected, analyzed, and interpreted. A well-chosen design can minimize bias, control for confounding variables, and provide strong evidence for or against a hypothesis. In contrast, a poorly chosen design can lead to misleading conclusions and wasted resources. Understanding the differences between cohort and case control studies helps researchers select the most appropriate approach for their research question, ultimately improving the quality of evidence in healthcare. Cohort studies are observational studies where a group of individuals, known as a cohort, is followed over time to observe outcomes. The cohort is typically selected based on exposure status, such as smokers versus non-smokers, and monitored to see who develops the outcome of interest, like lung cancer. This design allows researchers to calculate incidence rates and directly assess the relationship between exposure and disease. Cohort studies can be prospective, looking forward in time, or retrospective, using existing records to look back. Case control studies are another type of observational study, but they work in reverse compared to cohort studies. Researchers start by identifying individuals with the outcome of interest, called cases, and those without, called controls. They then look back in time to determine exposure status. This design is especially useful for studying rare diseases or outcomes that take a long time to develop. Case control studies are efficient and cost-effective, but they rely on accurate recall or records of past exposures. Both cohort and case control studies can be designed prospectively or retrospectively. In prospective studies, data is collected as events unfold, providing more control over data quality. Retrospective studies, on the other hand, use existing records or participant recall, which can introduce bias but are often faster and less expensive. Understanding the timing of data collection helps researchers weigh the trade-off between accuracy, cost, and feasibility when planning their study. Imagine a group of 10,000 adults is followed for 20 years to see who develops heart disease. At the start, researchers record lifestyle factors like diet and exercise. Over time, they compare the incidence of heart disease between those who exercised regularly and those who did not. This is a classic example of a cohort study, where exposure is measured before the outcome occurs, allowing for direct calculation of risk and incidence rates. Suppose researchers want to study a rare cancer. They identify 200 patients with the cancer cases and 400 similar individuals without it controls. They then look back at medical records to see who was exposed to a particular chemical. This approach is efficient for rare diseases, as it focuses on cases and controls rather than following a large population over time. The odds of exposure in cases versus controls are compared to estimate the association. Cohort and case control studies differ in how participants are selected and how data is collected. Cohort studies start with exposure and follow participants to see who develops the outcome, while case control studies start with the outcome and look back to assess exposure. This fundamental difference affects the types of questions each design can answer and the potential biases involved. Understanding these distinctions is vital for interpreting research findings accurately. Cohort studies offer several advantages. They allow for direct measurement of incidence and risk, can assess multiple outcomes from a single exposure, and are less prone to certain types of bias, such as recall bias. Because exposure is measured before the outcome, temporal relationships are clearer. These strengths make cohort studies particularly valuable for studying common diseases and exposures. Despite their strengths, cohort studies have limitations. They can be time-consuming and expensive, especially for rare diseases that require large sample sizes and long follow-up periods. Loss to follow-up can introduce bias, and changes in exposure status over time can complicate analysis. Researchers must carefully plan and manage cohort studies to minimize these challenges and ensure valid results. Case control studies are particularly efficient for studying rare diseases or outcomes with long latency periods. They require fewer participants and resources than cohort studies, making them more feasible for certain research questions. By focusing on cases and controls, researchers can quickly gather data and analyze associations, even when the outcome is uncommon in the general population. Case control studies have their own set of limitations. They are more susceptible to recall bias, as participants may not accurately remember past exposures. Selection of appropriate controls can be challenging, and the design does not allow for direct calculation of incidence or risk. These factors must be considered when interpreting the results of case control studies. Cohort studies are ideal when the exposure is rare but the outcome is common, or when researchers want to study multiple outcomes from a single exposure. They are also preferred when accurate measurement of exposure is possible at the start of the study. For example, investigating the long-term effects of a new medication in a large population is well suited to cohort design. Case control studies are best used when the outcome is rare or takes a long time to develop. They are also useful when the exposure data is available retrospectively, such as from medical records. For instance, studying the association between a rare cancer and environmental exposures is more practical with a case control design due to the efficiency in identifying cases. While cohort studies are less prone to recall bias, they can still be affected by other types of bias. Selection bias can occur if the cohort is not representative of the general population. Loss to follow-up is another concern, as participants who drop out may differ systematically from those who remain. Careful study design and follow-up procedures are essential to minimize these risks. Case control studies are particularly vulnerable to recall and selection bias. Cases may remember exposures differently than controls, especially if the exposure is well known or stigmatized. Selecting appropriate controls is critical. They should be similar to cases in all respects except for the outcome. Matching and careful control selection can help reduce bias, but some risk always remains. Cohort studies, the strength of association between exposure and outcome is typically measured using relative risk (RR) or risk ratios. This statistic compares the risk of the outcome in the exposed group to the risk in the unexposed group. A relative risk greater than 1 suggests a positive association, while less than 1 suggests a protective effect. This direct measurement is a key advantage of cohort studies. Case control studies use odds ratios (OR) to estimate the association between exposure and outcome. The odds ratio compares the odds of exposure in cases to the odds in controls. While odds ratios can approximate relative risk when the outcome is rare, they are not the same. Understanding this distinction is important when interpreting findings from case control studies. Confounding occurs when a third variable influences both the exposure and the outcome, potentially distorting the true association. Both cohort and case control studies must address confounding through careful design and statistical analysis. Techniques such as matching, stratification, and multivariable adjustment are commonly used to control for confounders and improve the validity of study results. Matching is a technique used in case control studies to ensure that cases and controls are similar with respect to certain confounding variables, such as age or gender. By matching cases and controls on these factors, researchers can reduce the impact of confounding and isolate the effect of the exposure of interest. However, overmatching can also be a concern, potentially masking true associations. Critical aspect of cohort studies is follow-up. Researchers must track participants over time to accurately measure outcomes. High follow-up rates are essential to minimize bias and ensure that the results are representative. Strategies such as regular contact reminders and incentives can help maintain participant engagement and reduce loss to follow-up. Both cohort and case control studies rely on accurate data collection. Sources may include medical records, interviews, questionnaires, or laboratory tests. The choice of data source affects the quality and reliability of the study. Prospective data collection generally provides more accurate and complete information, while retrospective data may be limited by missing or inaccurate records. Ethical considerations are paramount in all clinical research. Both cohort and case control studies must ensure informed consent, protect participant privacy, and minimize harm. Researchers must also be transparent about potential conflicts of interest and ensure that their studies are approved by relevant ethics committees. Maintaining high ethical standards builds trust and credibility in research findings. Determining the appropriate sample size is crucial for both cohort and case control studies. Too small a sample may lack the power to detect meaningful associations, while too large a sample can waste resources. Researchers use statistical methods to estimate the required sample size based on expected effect sizes, outcome frequencies, and desired confidence levels. One of the strengths of cohort studies is their ability to establish temporal relationships between exposure and outcome. By measuring exposure before the outcome occurs, researchers can more confidently infer causality. Case control studies, by contrast, are less able to establish temporality, as both exposure and outcome have already occurred at the time of study. Cohort studies are particularly useful when researchers want to study multiple outcomes from a single exposure. For example, following a group of smokers can reveal not only lung cancer risk but also risks for heart disease and stroke. Case control studies, on the other hand, are more efficient for studying multiple exposures related to a single outcome. Cost and feasibility are important considerations in study design. Cohort studies often require more time and resources due to long follow-up periods and large sample sizes. Case control studies are generally more cost-effective and quicker to conduct, making them attractive for certain research questions. Balancing scientific rigor with practical constraints is key. Many landmark studies in medicine have used cohort and case control designs. For instance, the Framingham Heart Study is a classic cohort study that identified major cardiovascular risk factors. The association between smoking and lung cancer was first established through case control studies. These examples highlight the impact of well-designed observational studies on public health. Interpreting the results of cohort and case control studies requires careful consideration of study design, potential biases, and confounding factors. Researchers must assess the strength and consistency of associations, the presence of dose-response relationships, and the plausibility of findings. Critical appraisal skills are essential for drawing valid conclusions from observational research. Transparent reporting is vital for the credibility of clinical research. Guidelines such as STROBE, strengthening the reporting of observational studies in epidemiology, provide checklists to ensure that cohort and case control studies are reported clearly and completely. Adhering to these standards helps readers assess the validity and applicability of study findings. In the hierarchy of clinical evidence, cohort and case control studies are considered observational designs, ranking below randomized controlled trials but above case reports and expert opinion. While they cannot prove causality as definitively as randomized trials, they provide valuable evidence, especially when trials are not feasible or ethical. Randomized controlled trials, RCTs, are considered the gold standard for establishing causality, but they are not always possible. Observational studies like cohort and case control designs fill important gaps, allowing researchers to study exposures that cannot be randomized, such as smoking or environmental factors. Each design has its place in the broader landscape of clinical research. Conducting cohort and case control studies involves practical challenges, such as recruiting participants, ensuring data quality, and managing logistics. Researchers must anticipate and address these issues to maintain study integrity. Collaboration among multidisciplinary teams, including statisticians, clinicians, and data managers, is often essential for success. Advances in technology such as electronic health records and big data analytics have transformed the way cohort and case control studies are conducted. These tools enable researchers to access larger datasets, improve data accuracy, and perform more sophisticated analyses. Embracing technology can enhance the efficiency and impact of observational research. Cohort and case control studies are conducted worldwide, providing insights into diverse populations and health issues. International collaborations can enhance the generalizability of findings and address global health challenges. Sharing data and harmonizing methods across countries are important steps toward advancing clinical research on a global scale. Future of cohort and case control studies lies in integrating new technologies, improving data sharing, and developing innovative study designs. Personalized medicine, genomics, and real-time data collection are opening new avenues for observational research. As these fields evolve, researchers must continue to adapt and refine their methods to answer complex clinical questions. Recap: cohort and case control studies are essential tools in clinical research. Cohort studies follow groups over time from exposure to outcome, while case control studies start with the outcome and look back at exposures. Each design has unique strengths and limitations, and the choice depends on the research question, available resources, and ethical considerations. Selecting the appropriate study design is a critical step in clinical research. Researchers must consider the frequency of the outcome, availability of exposure data, resources, and potential biases. Consulting with experts and reviewing the literature can help guide this decision, ensuring that the chosen design will yield meaningful and reliable results. Well-designed cohort and case control studies have had a profound impact on public health. They have identified risk factors for major diseases, informed prevention strategies, and shaped clinical guidelines. Their contributions underscore the importance of rigorous observational research in improving health outcomes worldwide. Common pitfalls in cohort and case control studies include inadequate control of confounding, selection bias, and misclassification of exposure or outcome. Awareness of these issues and proactive planning can help researchers avoid them. Regular training and quality assurance measures are also important for maintaining high standards in research. Collaboration is key to successful clinical research. Cohort and case control studies often require input from epidemiologists, clinicians, statisticians, and data managers. Working together ensures that studies are well-designed, data is accurately collected, and analyses are robust. Teamwork enhances the quality and impact of research findings. Effectively communicating the results of cohort and case control studies is essential for translating research into practice. Clear, concise reporting helps clinicians, policymakers, and the public understand the implications of research findings. Visual aids, summaries, and open-access publications can enhance the reach and impact of clinical research. Clinical research is a dynamic field, and continuous learning is vital. Staying updated on new methodologies, technologies, and ethical standards ensures that researchers can design and conduct high-quality studies. Attending conferences, participating in workshops, and engaging with the research community foster ongoing professional development. For those interested in learning more about cohort and case control studies, numerous resources are available. Textbooks, online courses, and professional organizations offer in-depth information and training. Exploring these resources can deepen your understanding and enhance your ability to conduct and interpret clinical research. In conclusion, understanding the differences between cohort and case control studies is fundamental for anyone involved in clinical research. Both designs have unique roles, strengths, and limitations. By selecting the appropriate study design and applying rigorous methods, researchers can generate valuable evidence that advances medical knowledge and improves patient care.